students, today I am going to discuss linkage and recombination. This concept is included in the chapter. This is principles of inheritance and variation. In fact, in this chapter we have studied about what are the basic laws of Mendel. So T.H. Morgan in 1910, T.H. Morgan, that is Thomas Hunt Morgan, in 1910 explain this process, that is what is the pattern of the genes which are located on the chromosomes, and this pattern of the genes he has explained on the X chromosome. Now let us see this cross A. In this cross he has selected a female which is this is the female parent and this is the male parent. So as we know that Drosophila is a unisexual organism, so here the female is having X and X chromosome and male is having X and Y chromosome. Now let us see what are the patterns of the genes he has selected. Let us see, this is the yellow color. This is the yellow bodied and this is the white eyed. These two genes, this is the white eyed. So if I have to label it, I can label this as Y yellow bodied and W white bodied. Now if I have to cross this with, see this is brown bodied and this is red eyed. These are the wild mutant and we can call this as Y plus and W plus. So when this cross was made in F1 generation, see what happens. These two chromosomes, they will separate independently. These two chromosomes will separate independently. In F1 generation, what we get is this progeny. So these are the F1. This is Y, this is W. So Y, W, and here we can say Y plus and W plus. So this is Y plus, W plus, and here this is Y and W. Okay, so this is F1. Now Mendel has continued his experiment by intercrossing the F1 hybrid. Now intercrossing the F1 hybrid, we are seeing this chromosome. So let us see what happens in this. This is one arm of this chromosome and this is its another arm, that is we call it as the sister chromatid. This chromosome, this is its one arm and this is its another arm. Clear? Now this is Y, this is W. So I will label this as Y, W, Y, W. Now this chromosome, it is having Y plus, this I will say it is Y plus, and this I will say it is W plus. Likewise, this is its sister arm, so we can say it is W plus, and this is Y plus. So what happens? There is a crossing over between these two non-sister chromatid, and this crossing over, it is in the meiosis 1. So it is exactly what happens. This chromosome will remain, this arm will remain as such. This arm, there occurs crossing over. In this arm also there occurs crossing over, but this arm remains as such. So what is the pattern now? This is having Y, this is having W, this is having Y plus, and this is having W, this is having Y, this is having W plus, this is Y plus, this is W plus. This is YW, this is Y plus W, this is Y, this is W plus. So I am just going to repeat this again. This is F1. In F1 there is YW and Y plus W plus. These are the full chromosomes. So when we are saying this as female, what happens? This chromosome is having these two chromatid. This chromosome is having these two chromatid. There occurs the process of crossing over between non-sister chromatid. So as a result of this, this part, it is like this, and this part, it is like this. So here in this chromosome, one arm is having YW and another arm of this chromosome is having Y plus and W. Similarly, in this chromosome, one arm is having Y and W plus, and another arm is having Y plus and W plus. Now, because this is male, so there is no, you can say, crossing over, as these genes which we are discussing, they are located on the X chromosome. So this is, before we go across for F2, this is the final situation. Now, in this, if we make a result for F2, this is F2, one thing, that is, we have to take this chromosome and cross it with all the four arms, for all the four chromatids of the female. This chromosome, see, this is the chromosome maternal, this is the paternal X, maternal X, paternal X, maternal X. Likewise, I have made all the four combinations with X chromosome of the male. Now, for the genes, this is Y with W, and this is Y and W. So you can see Y and W, both here we are having in the parental type, and this is what we are saying, it is yellow bodied with white eyes. So we can say here yellow bodied plus white eyed. Now we come to the second cross. This is YW and this is Y plus and W. So for Y plus, this is our Y plus and this is our W. Likewise with this, this is Y and W. So here, what is the new thing? This Y and W are the parental feature, but this is the new thing, that is Y plus. So Y plus is red bodied and this red bodied is, we call it as the wild thing. So this is wild. Now the third chromatid with this X chromosome, here you have yellow. Yellow means it is Y, and the second gene, it is black. Black means W plus. The parent is having, this X is having green, that is Y, and the red, that is the W. Now here also you can see there is Y and Y are parental. W is, this is a white eye, this is parental, but here you have white plus. White plus means this is red eye. So we can say it is a wild character, that is red eyed. Now with the fourth chromosome, this fourth chromosome is having, this is Y plus, this is W plus. Then this with this is Y and this one is the W. So this Y plus and W plus, that is both are the wild thing, like this. Now in another, if we have to take the cross with the Y chromosome, so here you can see this is the X maternal, X maternal, X maternal, again X maternal. This is the Y paternal, Y paternal, Y paternal, Y paternal. Now in Y paternal we don't have to show any genes because Y chromosome is not linked with any genes. Now let us see this, what happens in X. This is Y, this is W, this is red, so we can say this is red, this is red, this is black, this is black, this is blue. So from gene point of view, if you have to see, this is green, green means Y, W means this black, this is blue, this is Y plus, and red is W, this is W, Y plus, this is W plus. So here Y and W, these are parental, so we can say it is yellow and white, yellow, white. This is again Y is yellow and W is for, this is W plus. Yes, it is W plus. W plus is red-eyed. Red-eyed means white. Now here it is Y plus, so we can also say it is white. Here also it is white. Now this is the normal cross, what he is expecting, it was expected by Morgan. But in this cross, this was not the, you can say, result. Here the parental type was very high, that is 97.8% are parental, whereas the recombinant is only 1.3%, so it is 97.7%, this is 1.3%. What does it mean? Recombinant means when there is some new character in the sense that in the progeny None of it is with the yellow bodied with white eyed or the brown bodied with, this is our cross of brown bodied with red eyed. So in none of this, if you can say the yellow bodied and white eyed, brown bodied with red eyed, this is white, but some new characters are there, like yellow and white, yellow and red. This is none of the parent is having yellow and red. Similarly, none of the parent is having this combination, that is blue and this is Y plus and W. Similarly, none of the parent is having Y plus and W. So these are called as recombinant. So in this result, he obtained parental type as 97.7%. 98.7% and the recombinants were 1.3%. This was the first cross in which we can say that Y and W genes, they are very closely present. दोनों बिल्कुल पास में हैं, इसलिए इनके सेपरेशन के chances of separations are very low. Or we can say these genes are linked genes. ठीक है, here we come the word that is linked. Linked का मतलब है दोनों पास में हैं. The same thing we can say in another cross, that is cross B. In this B cross, what was the characteristic he has selected was if I say it is cross B. So cross B, it is a chromosome where two genes were selected, but these two genes were distantly placed. Suppose this is W gene and this is another gene, that is miniature wing gene. So you can see the distance between these two genes. So this was taken as the female. Now if I have to make a cross like this, the process will be same, but the frequency of the result is different. This is W plus and this is say M plus. So here again these genes are located on the X chromosome. Now in the F2 result, or for the result of F2 generation, if you see the result of F2 generation, he found that the parental type and the recombinant type, it is different than what he has expected in the cross 1. This is 37.2% recombinant and the parental type is 62.8%. This difference itself indicates that the distance between W and M, it is quite far, while Y and W, they are closely packed, they are closely present. So when the genes are closely present with respect to each other, we call this as linkage. While these genes, they are not closely present, they are distantly present, so we call they are not linked gene. So when we are saying the distance between two adjacent genes, this concept was described by Alfred Stewart. This Stewart was a scientist, was a student of the T.H. Morgan. So this, he has used the distance between two genes, and this concept was very useful in gene mapping, which was very useful in the human genome project. So as such, this process, it is linkage and crossing over, linkage and recombinant. Recombinant means that there are two new characters which were neither resembling with any of the parent. If we discuss here the recombinant will be W plus W and M plus a new character, or W plus or M the new character. So this is what it is called as recombinant. So this concept was explained by Morgan in the Drosophila.